I'm Dr. Malarkodi, Professor and Head, Department of Pharmacognosy, School of Pharmaceutical Sciences, Vistas, going to discuss about preliminary phytochemical screening of crude drugs. The contents are as follows. We are going to see what do you mean by the term phytochemicals, aim and principle of the phytochemical screening, classification of phytochemicals, advantages of phytochemical screening for carbohydrates, alkaloids, and saponins. Introduction. What do you mean by the term phytochemicals? The biologically active compounds present in the plant parts are called as the phytochemicals. These phytochemicals are derived from various parts of the plants, such as leaves, flowers, seeds, barks, roots, and pulps. They serve as a raw material base for the elaboration of more complex semisynthetic chemical compounds. Medicinal plants contain secondary metabolites like alkaloids, flavonoids, saponins, tannins, and glycosides, which are used against different diseases and relieve pain. Herbal medicines are mostly used because they are easily available, have very less toxicity, are inexpensive, and have few side effects. Aim and principle to carry the phytochemical screening of the crude drugs. The main principle is to identify the phytoconstituents by preliminary phytochemical screening. Phytochemicals, in the Greek language, the term phyto means plant, refers to the biologically active, naturally formed chemical compound in the plants. The phytoconstituents work with nutrients and fibers to form an integral part of defense system against various diseases and stress conditions. The preliminary phytochemical screening involves testing of different plant extracts for their content of different classes of compounds. It can be done by qualitative chemical analysis using specific reagents for specific constituents. Classification of phytochemicals. The phytochemicals are basically divided into two groups. They are primary metabolites and secondary metabolites. What do you mean by the term primary metabolites means the essential molecules produced by organisms that are directly participating in the growth of plant development and reproduction are called as the primary metabolites. The secondary metabolites are organic compounds produced by living organisms like plants, fungi, and bacteria that are not directly involved in the primary growth, development, or reproduction of the plant. Classified according to their functions in plant metabolism as primary metabolites, examples are carbohydrates, amino acids, proteins, etc. The secondary metabolites found in the plants are alkaloids, phenolic compounds, flavonoids, tannins, glycosides, terpenoids, saponins, etc. Advantages of the phytochemicals. The preliminary screening of the phytochemicals is a valuable step in the detection of bioactive principles present in the medicinal plants. Subsequently, they may lead to drug discovery of development. Findings from the laboratory studies have shown that phytochemicals have the potential to stimulate the immune system. Diets rich in certain phytochemicals can reduce the risk of several common diseases. The requirements and procedures to carry out the phytochemical screening. The basic chemicals, funnel, measuring cylinder, filter paper, test tube holder, and water bath. Procedure. How to prepare the extraction of the crude drug. Take about 50 gram of the powdered crude drug and macerate it with 500 ml of ethanol in a closed conical flask for about 24 hours. Then occasionally shake and allow it to stand for 18 hours. After filtration, evaporate the filtrate to dryness in a dark, a flat bottom shallow dish. Preparation of the test solution. Take about 500 milligram of extract and dissolve it in 100 ml of ethanol. Stir the solution till the extract is completely soluble in ethanol. The sample solution then subjected to various qualitative tests to reveal the presence or absence of common phytochemicals. First, we will move on to the test for carbohydrates. Molish test. Place 2 ml of a known carbohydrate solution in a test tube. Add about 1 drop of Molish reagent. The chemical name of Molish reagent is 10% of alpha naphthalene in ethanol. Pour about 1 to 2 ml of concentrated sulfuric acid down the sides of the test tube so that it forms a layer at the bottom of a tube. Based on the degradation of the carbohydrate by sulfuric acid to produce an aldehyde, either a furfural or a derivative, it condenses with a phenolic structure resulting in a red or purple colored compound. Failings test. To 1 ml of failing solution A, which is called as the aqueous solution of copper sulfate, add 1 ml of failing solution B, which is called as the solution of potassium tartrate. Add 2 ml of the sugar solution, mix well, and then boil. A red precipitate of cuprous oxide that forms at the end of the reaction can be seen. In the case of reducing sugars, the presence of alkali causes extensive enolization, especially at high pH and temperature. This leads to a higher susceptibility to oxidation reaction than neutral or acidic pH. The sugars therefore become potential agents capable of reducing cuprous to cupric, and Fehling's test is commonly used test for detection of reducing sugars. A reducing sugar is any sugar that in a solution has an aldehyde or a ketone group. Barford's test. To about 1 to 2 ml of Barford reagent, add an equal volume of sugar solution. Boil for 5 minutes in a water bath and allow to stand. A brick red cuprous oxide precipitate, if reduction has taken, will be observed. Barford reagent is cupric acetate in acetic acid, which is slightly acidic and is balanced so that it can only be reduced by the monosaccharides. Shalifanoff's test. Heat about 1 ml of sugar solution with 3 ml of Shalifanoff's reagent, which is about 0.5 gram of resorcinol per liter with 10% hydrochloric acid in boiling water. In less than 30 seconds, a red color must appear for ketosis. Upon prolonged heating, glucose will also give an appreciable color. Shalifanoff's test distinguishes between the aldose and ketose sugars, and ketoses are distinguished from aldoses through their ketone aldehyde function. If the sugar contains a ketone group, it is a ketose, and if contains an aldehyde group, it is an aldose. This test is based on the fact that when heated, ketoses are more rapidly dehydrated than aldoses. Biol's test. To about 5 ml of Biol's reagent, add 2 to 3 drops of sugar solution and boil. Upon boiling, note the green-blue color formed. Biol's test is used to determine the presence of pentoses. The components of this reagent are resorcinol, hydrochloric acid, and ferrichloride. In this test, the pentose is dehydrated to form furfural, and the solution turns bluish color, and a precipitate may form. Iodine test. To about 2 to 3 ml of polysaccharide solution, add 1 to 2 drops of iodine solution. Iodine is a very good indicator of presence of starch. Iodine solution is dissolved aqueous solution of potassium iodide, which reacts with starch to produce a blue-black color. Next, we will go on for the test for alkaloids. A known quantity of the extract, 0.1 milligram, was added to 6 ml of dilute hydrochloric acid and boiled. After boiling, it is cooled and filtered. The filtrate was divided into 4 portions and subjected for the following test. To the first portion, 2 drops of drag and drop reagent is added. Drag and drop reagent chemical name is potassium bismuth iodide. The formation of an orange-red precipitate indicates the presence of alkaloids. Second test, Mayer's test. To the second portion, 2 drops of Mayer's reagent is added. Mayer's reagent is chemically called as potassium mercuric iodide. A creamy white precipitate indicates the presence of alkaloids. It is the most commonly confirmed test used for the presence of alkaloids. The solution should be added distinctly in acidic solution of the alkaloid. Only a few drops of the reagent should be used, and the solution should not contain acetic acid or alcohol. Next comes the Wagner's test. To the third portion, 2 drops of Wagner's reagent is added. Wagner's reagent is chemical name is iodine potassium iodide solution. A reddish brown precipitate indicates the presence of alkaloids. Hager's reagent. To about 2 to 3 ml of the filtrate, few drops of Hager's reagent, which is saturated solution of picric acid, was added. Appearance of yellow color precipitate revealed the presence of alkaloids. It can be noted that alkaloid precipitate reagents may give a precipitate with other plant constituents. They also give false positive reaction such as proteins, amino acids, tannins, volatile oils, and some flavonoids and coumarins, which are all nothing but oxygenated natural products. Therefore, it is necessary to get rid of such constituents when testing for the presence of alkaloids in plants. Then test for saponins. It is also called as the foam test. A few milligrams of the test residue was taken in a test tube and shaken vigorously with a small amount of sodium bicarbonate and water. Appearance of stable, characteristic honeycomb-like froth indicates the presence of saponins. Thanks.